Hi everyone, I'm Sanjay Lad. Welcome to one more video of SPL BIO-TONIC. In today's session, we'll talk about a new chapter called as Molecular Basis of Inheritance. And today we'll start with Central Dogma of Molecular Biology. Students, I request you all to like my channel, share it and subscribe it. And don't forget to press bell icon. And let us start with this concept of Central Dogma of Molecular Biology. Now, central dogma of molecular biology basically talks about the flow of information from DNA to proteins. Now, we know that DNA is made up of nucleotides, RNA is also made up of nucleotides, whereas proteins are made up of amino acids. So, students, DNA is a polymer of deoxyribonucleotides, RNA is made up of ribonucleotides, whereas proteins are made up of amino acids, which are joined together by peptide bonds. Now, formation of DNA to DNA, formation of DNA to DNA is called as replication. It is also called as duplication. It is called as DNA replication or DNA duplication. Formation of RNA from DNA is called as transcription, whereas formation of proteins from RNA is called as translation. Now, students, if you look at the language of DNA and RNA, both have language of nucleotides. So conversion of one language to same language, that is termed as transcription. One language to same language is called as transcription, whereas one language to other language, that is language of nucleotides to language of amino acids, is called as translation. Now here, translation is also called as protein synthesis. Translation is also called as protein synthesis. So what this central dogma of molecular biology says, that there is a flow of information from DNA to RNA and RNA to protein, and this process is never reversible, or the process is not reversed. So there is always flow from DNA to RNA, RNA to proteins. Now in some cases where, like in case of retroviruses, RNA information can be converted to DNA. That process called as reverse transcription. Even at some latter part of, say, molecular biology and biotechnology, we'll be talking about cDNA technology, that is, say, combined DNA technology, where we'll be talking about this same process, that is, conversion of mRNA into DNA. So, time being, let us understand this central dogma here. Now, in case of eukaryotic cell, as there is a nucleus, process of replication and transcription occurs inside the nucleus, whereas the process of translation occurs inside the cytoplasm. If you compare this eukaryotic cells now with prokaryotic cells, the processes are same, but there's a difference in the site of these processes. Now, there is a flow of information from DNA to RNA and RNA to proteins in prokaryotic cells also. But in case of prokaryotic cells, there is no nucleus. So therefore, replication, transcription and translation, all these three processes occurs inside the cytoplasm only. So in case of eukaryotic cell, replication, transcription occurs inside the nucleus, whereas translation occurs inside the cytoplasm. Whereas in case of prokaryotic cell, all these three processes occurs inside the cytoplasm only. So students, I hope you are understanding the central dogma of molecular biology. So I request you all to like my channel, share it, subscribe it, and don't forget to press bell icon. Thank you.